Imagine a grain turning into cheese or a jar of pickles, all thanks to tiny bacteria working silently behind the scenes. Fermentation is a natural biochemical process in which microorganisms convert complex organic molecules into simpler compounds, often producing energy for themselves and altering the food matrix. In food systems, bacteria are the primary agents that initiate fermentation by metabolizing sugars present in grains or fruits. The metabolic pathways employed by these microbes typically involve glycolysis followed by lactic acid production or ethanol synthesis, depending on oxygen availability. Lactic acid bacteria consume glucose and release lactate, which lowers the pH of the surrounding environment. The drop in acidity inhibits spoilage organisms, while simultaneously creating a distinct sour flavor profile that is characteristic of products such as yogurt or sauerkraut. In addition to acidification, bacterial fermentation can produce gases like carbon dioxide, which contributes to the texture and aeration seen in bread or fermented beverages. The enzymatic activity of bacteria also breaks down complex carbohydrates into simpler sugars, making nutrients more bioavailable for both humans and the microbes themselves. Over time, these biochemical transformations stabilize the food by reducing water activity and creating a hostile environment for pathogenic bacteria. The resulting fermented product often exhibits enhanced flavor complexity, improved shelf life, and sometimes added nutritional benefits such as probiotics. Thus, bacteria serve as the engine of fermentation, converting raw substrates into stable, flavorful foods through a series of well-orchestrated metabolic steps. Pause 0.8. Lactic acid bacteria (LAB) are a diverse group of microorganisms that convert sugars into lactic acid during fermentation. 3 families dominate industrial fermentations: Lactobacilliaceae, Streptococcaceae, and Pediococcacea, each with distinct metabolic traits. Members of Lactobacillus produce homofermentative pathways that yield primarily lactic acid from glucose, creating a stable acidic environment. Streptococcus species often exhibit heterofermentative metabolism, generating lactic acid along with small amounts of carbon dioxide and ethanol. Pediococcus strains are known for their salt tolerance, allowing them to thrive in high-salt vegetable fermentations such as kimchi. In dairy products, Lactobacillus delbrueckii subs bulgaricus collaborates with Streptococcus thermophilus to acidify milk into yogurt. Vegetable fermentations like sauerkraut rely heavily on Pediococcus pentasacilus, which maintains pH stability during prolonged pickling. Grain-based sourdough starters frequently contain Lactobacillus sanfrancesensis, a species that tolerates high acidity and contributes to the Breda Eurostrade flavor profile. The geographic distribution of these LAB families reflects local dietary habits. For example, Streptococcus thermophilus is prevalent in European dairy traditions. Understanding the specific metabolic pathways of each family allows food technologists to predict fermentation outcomes and optimize product quality. Pause 0.8. Lactic acid bacteria initiate fermentation by consuming hexose sugars through the glycolytic pathway. In this first stage, glucose is phosphorylated and rearranged to produce 2 molecules of pyruvate while generating ATP. The key redox reaction that follows transfers electrons from NAD to pyruvate, forming lactate and regenerating NAD+. Lactate dehydrogenase catalyzes this conversion by binding both pyruvate and the cofactor NAD at its active site. Structural studies reveal a Rossmann fold that positions the nicotinamide ring for efficient hydride transfer. The enzymia Eurostrade-S catalytic residues stabilize the transition state, lowering the activation energy of lactate formation. As lactate accumulates, it lowers the pH of the surrounding medium, creating an environment that favors further bacterial growth. This acidification also acts as a preservative by inhibiting spoilage organisms during food processing. The overall stoichiometry of the reaction is glucose A circumflex 2 pyruvate plus 2 ATP A circumflex 2 lactate plus 2 NAD+. Understanding this pathway allows food technologists to manipulate fermentation parameters for desired flavor and texture profiles. The rapid regeneration of NAD+ ensures that glycolysis continues unabated, sustaining the bacterial energy supply. Consequently, lactic acid bacteria produce high concentrations of lactate in a matter of hours, which is essential for the characteristic sour taste of fermented dairy products. Pause 0.8. Pause 0.8. Kombucha fermentation begins when a sweet tea solution is inoculated with a symbiotic culture of bacteria and yeast, commonly called a SCOBY. The bacterial component, often gluconacetobacter xylanus, attaches to the surface of the liquid and secretes cellulose fibers that form the gelatinous layer. Yeast cells metabolize sucrose into ethanol and carbon dioxide, creating an anaerobic microenvironment favorable for bacterial growth. As yeast produces ethanol, Acetobacter oxidizes it further into acetic acid, which lowers the pH and imparts a characteristic tangy flavor. Concurrently, bacteria convert residual sugars into gluconic acid and other organic acids, contributing to the complex sour profile of kombucha. The interplay between yeast respiration and bacterial oxidation establishes a dynamic equilibrium that balances acidity, sweetness, and carbonation. Over several days, the SCOBY thickens, and the liquid develops subtle fruity notes derived from secondary metabolites produced by both microbes. Time-lapse footage reveals this gradual transformation, illustrating how microbial cooperation yields a beverage rich in diverse flavor compounds. A schematic of bacterial-yeast interactions highlights the directional flow of substrates: sucrose, ethanol, acetic acid, and gluconic acid. Understanding these mechanistic steps allows brewers to manipulate conditions such as temperature or sugar concentration to tailor taste profiles. Pause 0.8. Bacteria convert sugars into organic acids, gases, and enzymes during fermentation, each contributing distinct sensory changes. The accumulation of lactic acid lowers pH, which tightens protein networks in dairy products, yielding a firmer curd texture. Acetic acid produced by acetic bacteria imparts sharp, vinegar-like flavor while also creating an environment hostile to spoilage microbes. Carbon dioxide released by yeast and some lactobacilli expands cell walls, producing the characteristic bubbles seen in sourdough bread. Enzymes such as proteases break down casein in cheese, releasing peptides that enhance umami notes and soften the crumb structure. The combined effect of acidification and enzymatic action extends shelf life by inhibiting pathogenic growth and slowing oxidative rancidity. Sensory panels often correlate higher acidity with increased tartness perception, while texture descriptors shift from soft to firm as pH drops. Bar graphs comparing titratable acidity across fermented products reveal that yogurt typically has the lowest pH among dairy items studied. In pickled vegetables, elevated lactic acid levels correspond to a crisp bite and reduced water activity, further preserving the produce. By mapping biochemical outputs to sensory attributes, producers can fine-tune starter cultures for desired flavor profiles and product stability. Pause 0.8. Bacteria are the engines of food fermentation. Lactic acid bacteria lower pH through lactate production. Acetobacter oxidizes ethanol into acetic acid for preservation. Kombucha trade as symbiotic cultures generate diverse acids and flavors. These biochemical activities shape the taste, texture, and longevity of fermented foods.